Good morning, everyone. So thanks for the introduction, Leslie. What I want to do today is to introduce you to Central Biotechnology Services, CBS, as we all like to call it. And I think it's fair to say that if you're doing a laboratory-based project in the school, you will be using our facility. I think that's almost a given because of the range of things that we offer. We're based within the Time Institute, the Institute for Translation, Innovation, Methodology and Engagement. So what CBS does is it offers and it serves as a place for the School of Medicine core research facilities. So we offer state-of-the-art technology platforms, particularly in the post-genomic and imaging areas. It's staffed and managed by a team of, we're actually sort of getting towards 18 people now once everyone is appointed, and we offer a range of technological and project management expertise. One of the key things about CBS is that we have additional key input from lots of academic researchers. I mean, I put 6+ academic researchers down there just to remind me to say that there are actually sort of 6 or 7 academic researchers that essentially work all the time on a weekly basis with CBS. Beyond that, and offer their academic expertise. Beyond that, there's a whole range of other researchers that kindly give us their time and expertise to help with certain specific projects. So we offer a range of services across a broad spectrum of workflows, and one of the key things is that we aim to be a sustainable resource. So your grants, your supervisors will pay to use our facilities. And because of that, if you use our facilities, I think it's very important to sort of keep in the loop with your supervisor and let them know what you're doing or what you're planning to do, because ultimately things need to be paid for. So our role, and this is sort of our broad role, not just within the School of Medicine, is to offer services, technological support, training, to enter into extended collaborations and offer academic expertise or consultancy, certainly for School of Medicine researchers at all levels, Cardiff University researchers, but we also work very closely and have contracted projects with external not-for-profit organisations, particularly the NHS and other universities, and also commercial organisations ranging from very, very small startup companies to small and medium-sized enterprises to big sort of global companies such as GE Healthcare, and we do a lot of work for GE Healthcare, just as an aside. So in order to do this external work, we work to very high quality standards, and we're ISO 9001 accredited. We do things in a very, very careful and methodical way, and I think for this audience that's all I really wanted to say. So what are the facilities that we have? I hope you can read this slide, and I'll come back to these technologies in a little bit more detail sort of shortly. But we have a range of genomics technologies, proteomics technologies, a BioCore facility, a platform for high throughput assays. We also have a range of imaging facilities, sort of electron microscopy, histology facility, and also a facility for sorting cells and analyzing cells by flow cytometry. To go into these in a little bit more detail now, and you know, I could drone on and on here, so I'm really sort of just going to sort of give you a few headlines, and you've got these pretty pictures that you can perhaps aim to match up with the different technologies that we offer. We have an established DNA sequencing facility. This is standard Sanger sequencing. We're in the process of integrating with the NHS facility in order to offer a better resource here. And in the future, but a little way off yet, we're going to get into and be able to offer next generation sequencing facilities, but that's certainly about 12 months away. We have a range of microarray platforms, an Affymetrix facility, which is our oldest platform, a Torre facility, and Illumina facilities. And these technologies, they all have their strengths and their weaknesses, but you can use them to look at gene expression, to do SNP genotyping, maybe look at copy number variation, these sorts of things. Some of the technologies are better suited to messenger RNA, such as the Affy system. The Torre system, for example, is very well suited, and certainly the company promotes it very heavily for looking at microRNA. And we're actually the European Service Centre for this relatively new technology. So we work very, very closely with Torre Industries in order to deliver this service to researchers across Europe, but particularly within Cardiff. We have a very well-established qPCR facility, and Claudia within that facility will be able to offer you a considerable amount of expertise in planning your experiments to get the data that you want. And this is really sort of what we do with all of these facilities, is that we have technology experts that work with these technologies day in, day out. So, you know, what you need to be doing is at the point of inception, sort of designing your experiment, talking with these technologists in order to design your experiment the best way in order to produce the best data and then the best output. And we'll help you along that journey in order to get the best work done that is possible. So beyond your research group and your supervisory team, there's this really significant resource that can help you to conduct experiments using the platforms that we standardly offer. My own area is in proteomics, and we have facilities for identifying and quantifying proteins, either individual proteins from gels or looking at things more globally. And we have the mass spectrometry platform that we have to do this is a MALDI TOF instrument, and we also have a liquid chromatography system for separating out peptides, a Dionex system. There's a high specification sort of confocal scanning imager, a Typhoon system, and this can be used to look at fluorescence in gels. We have a Biocore facility. The Biocore instrumentation, it does surface plasmon resonance, and it's used to look in real time in a label-free way at macromolecular interactions. These might be protein-protein interactions or other interactions such as protein-DNA interactions. So a very, very useful resource. We have an MSD system, a mesoscale discovery system, and this is for biomarker detection, the protein detection, and it can be done sort of in a single assay sort of format or a multiple sort of assay format. And it's a high throughput technology, and there are panels that can be used. So, for example, you can take samples and look at a range of different cytokines and the amount of cytokines that are present within those samples. So that's one of the panels. There are a whole range of other panels. Come to talk with us if you want to know more about that technology. On the flow cytometry side, we have a range of benchtop instruments, and those of you from an infection and immunity background, but also perhaps other backgrounds, will be using these, I guess, fairly extensively within your studies. We've currently got 3 instruments, benchtop instruments for doing flow cytometry, and also an ARIA system, a relatively new system, certainly less than a year old, for cell sorting. We offer, as we do with all of our technologies, we offer training in these technologies so that you can work safely. Okay, and that's both safe to yourself and safe to the instrument, so that the technology doesn't get sort of damaged at all. In relation to flow cytometry, we will be offering specific training courses, which will be starting sort of very soon for how to use the flow cytometers safely. On the 1st of November, there's going to be a whole day training course from an external organisation, which is going to be focused in on really, rather than actually doing the experiments and actually running an instrument, how to design your experiment and the theory behind flow cytometry. And if you're interested in attending that whole day training course, it's going to cost your supervisor a small amount of money, but I think it'll be highly worthwhile, and please email us if you're interested in attending that course. We will be sending out circulars very shortly for that as well. Moving on to sort of imaging and histology, we have a range of different types of confocal microscopes that are that you can access through CBS, and this is run primarily by Chris George in the Heart Research Institute, and we have a range of sort of light microscopy solutions as well. We have an electron microscopy facility. It has one scanning electron microscope currently in operation, one TEM, one transmission electron microscope, and beyond that there's a second TEM that's going to be up and running fairly soon. In very close collaboration with the NHS, we also offer histology services, and you may need to avail yourself of these in your studies if that's what your project demands. So what else can we offer? Well, beyond those sort of platforms that we've got the range of platforms, a range of expertise. What we can also do is work with you potentially and with your supervisors to really sort of try and sort of tailor things and give you access either to other resources within our institute or to work with colleagues in other research institutes to enable you to have access to their resources and also to other Cardiff University facilities. Now, of course, this is something that you can do off your own back, but for example, it is very, very common for us to get an inquiry saying, I want to use this piece of equipment, it's not available in CBS, I don't know where to access it. And we can help you to locate where you might be able to access that piece of equipment. Or if that particular piece of equipment is not available, we may be able to come up with another solution. So we're very, very happy to work with you as facilitators for what you want to do. There's really one more thing that I wanted to say. We've got a range of sort of contacts up there. The key contact really is this CBS admin email address, and there's a phone number there. Certainly go to the website, okay, for CBS. Through the website, there's an online booking system. You can, as long as you're within Cardiff University, you can create a new account there. You can do that yourself. You don't have to come and see us. You can create a new account, and you can either use your own user details or make up some entirely new details. It's entirely up to you. Once you've created that account, you can book CBS equipment sat at home. You could book CBS equipment when you're on your holidays if you want, for when you come back. Beyond everything that I've said, the one thing I guess that I've missed out within these slides is that we are in the process of recruiting a small team sort of for CBS, sort of 2.5 individuals for bioinformatics. And please do come and talk to us if you have any queries or you feel that you might need any assistance in terms of data analysis support, bioinformatics support. If we can't directly help you, we may be able to facilitate some training elsewhere that will be able to help you get into your data, which for many of us is a key challenge, and I think it's one of the things that you shouldn't underestimate, that you'll need to spend really quite a lot of time with, really putting in the hours in order to train yourself to be able to deal with the data that you've got and to get as much out of the data as you possibly can. And we'll aim to help you on that journey. That's all I wanted to say. Thanks for your time.